type 2 diabetes. So increased cellular ACRP30 protein levels can be beneficial as a treatment for insulin-resistant diabetes in muscle, fat, and liver cells. Accordingly, I postulate the use of a RAAV vector plasmid in which 0.81 kilobase DNA fragment for human ACRP30 gene, therapeutic gene of interest, will be inserted and will be supplied in trans by the Rep2/Cap2 plasmid during the triple transfection for RAAV production. Conclusion: the ACRP30 gene will be the therapeutic gene of interest in our study, and we would like to insert it into the RAAV vector. After completion of triple transfection, RAAV variant particles containing the ACRP30 gene in the DNA and a Rep2/Cap2 capsid are created. Thereon we will inject into the bloodstream, intravenous or intraarterial or intramuscular, this recombinant adeno-associated virus into the central nervous system of an insulin-resistant diabetic patient, and wait for integration of the virus carrying the therapeutic gene of interest onto the specific site on human chromosome 19. The ACRP30 gene will be expressed from chromosome 19. There will be an increase in the cellular level of ACRP30 protein, and we would expect a decrease in endogenous glucose production without lowering peripheral glucose uptake, and thereby resulting in decreased blood glucose concentration. These steps will be beneficial in the treatment for insulin-resistant diabetes in muscle, fat, and liver cells, and will eliminate type 2 diabetes at the nuclear DNA, that is chromosomal level. HIV/AIDS. HIV/AIDS is the retroviral disease with a high fatality rate in humans. After reading a plethora of scientific publications and seeing the progress made to date, I have concluded to identify and devise an experimental design for its treatment. The HIV-1 integrase protein plays a crucial role in generating hydroxyl ends on the 3' end of the viral DNA after action of reverse transcriptase within the cytoplasm. Integrase also forms a complex with the viral DNA, and after transport into the cell nucleus, HIV-1 integrase acts as a catalyst in ligating viral DNA into host DNA at a random site on a random chromosome by the hydroxyl group on the viral DNA attacking the 5' phosphate end on the host DNA chromosome by formation of a phosphodiester bond. Now the viral DNA is fully integrated into the host DNA. An important area of research is the discovery and development of integrase inhibitors. Diketo acids are potent inhibitors of HIV-1 integrase that inhibit integration and viral replication in cells. A protein from Corynebacterium species, which is a diketo acid, is 2,5-diketo-D-gluconic acid reductase A, 277 amino acids long, which equates to 0.831 kilobase DNA or 30.77 kilodalton protein. This is a therapeutic gene of interest. Interestingly, this protein also catalyzes the conversion of 2,5-diketo-gluconic acid obtained by conversion of D-glucose using a prokaryotic microorganism to 2-keto-L-gluconic acid, which is then converted to L-ascorbic acid, vitamin C, in the human body. Accordingly, I postulate the use of a RAAV vector plasmid in which the 0.831 kilobase DNA fragment for 2,5-diketo-D-gluconic acid reductase A gene, the therapeutic gene of interest, will be inserted and will be supplied in trans by the Rep2/Cap2 plasmid during the triple transfection for RAAV production. Other promising methods for treatment of HIV/AIDS include administering of radiation, induction of apoptosis by use of executioner protein caspase-6, and possibly some method of insertion of PES protein sequence as a signal peptide for protein degradation onto the translated viral polypeptide after it exits the cell nucleus and exists in the cytoplasm. Conclusion: